Welcome back to PricePlow. We're here with Danielle Citrolo of Kyowa Hakko USA, the makers of Cognizin Citicoline. Danielle, welcome to the show. We have a lot of information coming about Cognizin, a new study that you want to talk about. But before we begin, I'd love to have you introduce yourself. And we've never done like a formal introduction to Cognizin Citicoline on this podcast. Most of our listeners should know what it is, but how about we give them the 1 minute elevator pitch on that as well. So thanks for joining us.
Thanks for having me, Mike and Ben. I'm Danielle Citrolo. I am the Vice President of Scientific and Regulatory Affairs at Kyowa Hakko USA. And one of our leading ingredients, we sell dietary ingredients, and one of our leading ingredients is Cognizin. And for those who may not know, Cognizin is, I think, one of the best nootropics out there. It helps with focus, attention, memory. It also has some increases in brain energy, and then it really helps overall brain support. And I think that's how it differentiates itself in the marketplace of nootropics, is that it helps in the now with that focus, attention, all those things you need. But it is truly a nutrient for your brain that does a lot of good for the structure, support, function. And then this new science that we have is like cutting edge Metabolomics, which is going to really show us all the good things that it's doing in our brain.
Excellent. Yeah, the one thing we always love talking about with Cognizin is that it's experiential. And we noticed this, the first video we did, it was before Ben was with PricePlow, it was like 2013. There is a pre-workout that's no longer on the market from a brand that's really well known. They came out with Citicoline, and it like knocked our socks off. And so we were like very excited to have an experiential ingredient. I think that's what a lot of people are looking for. And that's why we're seeing a lot of success with the ingredient and the way that you are continually, studying it. This is not the first study that you've had on this ingredient.
Absolutely not. And there's more to come. And in fact, my colleague, Katie Emerson, who's been on your podcast, I think, before,
Yep.
is actively working on new science. And we're always going to be coming out with new science and new benefits for this ingredient.
So we've talked about a lot of the research in the past. In humans, 250 milligrams and 500 milligrams have different results and both very successful doses and everything. What happened in this study? Can you just give us the whole rundown if you don't mind?
Yeah, absolutely. Well, I'll give you the higher overview, and then I think when we get into some of the nitty gritty, it's maybe better to share some of the slides and diagrams so you can see it, because it's a lot. But generally speaking, we can't easily study how basically what we're looking at with this new study is how citicoline impacts how our genes are expressed. So we have DNA, and DNA is kind of the code of, how our, cells can work. But mRNA is how it's expressed. So it's how the genes are coded. And it basically upregulates our proteins, our enzymes, various metabolic pathways. So it's like the expression of your DNA. And it's kind of like the footprint of when I ingest this is what my body does. This is how my DNA reacts. This is what's upregulated in my body. And these are the benefits, or, in some cases, not cognizant, but it could be negative benefits too, right? So it's really the expression, and it really tells us exactly the mechanism. So that's the science I'm going to get into. It's really cool. It's cutting edge science. I was at the American Society for Nutrition conference in June. There was a lot of metabolomics data. I really think ours really took the cake on it in terms of quality. So I was pretty proud of that. So if you want, I can just get into it, unless you wanted to kind of give any more detail or context. Yeah.
I guess when designing a study, you're kind of like, you either come with a hypothesis or perhaps a goal, something you want to say, something you want to know. Like, you're not just throwing money at something for no reason. So what were the reasons for even like going down this path?
Absolutely. Great question. So Cognizant has been studied in many different healthy subjects. We know the benefits I just mentioned: focus, attention, memory, all this health of the brain cells, right? And then there's also clinical data that shows it can help with, other diseases and stroke and things like that. But we really didn't know. We knew a general idea of how it worked, but this science is telling us exactly how it truly does work at the cellular level. Now, we can't take brain tissue out of humans. That's not ethical. So what we did was something really special, what's like called a triculture, and I'm going to get into it a little bit more, representing what would be like brain tissue. And then we ran the genetics on that triculture, which is what we would represent. But we also did something to check neuronal networking. Really, since we know it excites the neuronal network because it helps with neurotransmitters, we have the science to show focus and attention. We tested though and looked at that. So that's really cool.
Okay, awesome. So I guess let's get into it then. And then as you get into it, you'll probably want to explain the triculture model that you just discussed, because that's never been talked about here.
Yeah, definitely. I think I have a good slide, but
okay. Yeah. So for those listening on audio feeds, we'll do our best for you, but you're probably going to want to watch YouTube if you're really into this stuff.
So this is the why you were kind of asking about before. Why would we go after metabolomics research? Ultimately, we were trying to nail down and understand the various mechanisms behind Cognizant. The things we already know is we know it enhances phospholipids, which help with brain cell health, cell membrane turnover. We know that it can increase acetylcholine and dopamine, which help with that focus and attention. But the last one is the mitochondrial function, which we do have science showing we can increase ATP levels in the brain. But how exactly we've got to that, nobody really knew. So I think this new science actually does help us understand a little bit about that third leg and just overall some of the other mechanisms behind just long-term brain health support. So just to kind of explain, we're going to be talking about transcriptomics, which I quickly touched on earlier. And it's basically the expression of our DNA through RNA. RNA, when we look at RNA expression, it helps tell us what biosynthesis of enzymes is happening, what proteins are being expressed. It helps signaling and biochemical reactions throughout the body. So it's really like, speaking, kind of translating our DNA into action is what transcriptomics is. And it's really measuring mRNA expression. So I'm going to get into a little bit of, it's basically two different parts to this study. First is that we took immature pluripotent stem cells, as I described before. We have this great technology where we can use stem cells and differentiate them into whatever type of cell we want. And in this case, we differentiated them into 3 types of brain cells: neuron, microglia, and astrocytes. And we felt that, and together we made a culture that was basically equal parts of these 3 types of cells to represent what would be like brain tissue, since we can't extract that from humans. And right. We did 2 things with those cells. We ran them through a neuronal network with electrophysiology recording, looking at like neuronal excitatory network to see, are we exciting the neurons compared to like placebo? But then we also took those cells and we mashed them up and we ran them through mRNA sequencing to look at what the footprint of citicoline is as compared to placebo. But it's actually a 4-arm study, which is actually even more interesting, and I'll get into that in a minute. So one
quick question. So the triculture type study, is that like a standard that's accepted in the world out there?
Yes.
Okay,
it's definitely the most, you can do, there's other cells, but this is the highest. There are other types of neuronal cells, but these is the highest concentration in brain tissue. Mm-hmm. So that's why we chose that. And Perfect. Just a quick one on like, what are these? I think most people know neurons are the brain, your brain cells, but there's these other types of cells, and there's more than just this, but the 3 that we talk about are microglia and astrocytes. And microglia are kind of like the immune cells in our brain. And then astrocytes are supporting cells to the neuron. They provide structural support, metabolic support, which is a note because the metabolic support comes into some, and neuroprotective. So there's 3 different types of cells. I just wanted to share like, what are those cells, because I think it's important. So this was a 2-part study. We utilized the triculture cells. So it's 3 different types of neuron cells, and they were actually differentiated from immature pluripotent stem cells. So we have this great technology nowadays where we can use stem cells and differentiate them into any type of cell we want. We chose neurons, microglia cells, and astrocytes, as those is the most numerous amounts of types of neuronal cells in our brain. It would be most representing brain tissue. And we did two parts of the study. First is we put it through an electrophysiology test where it tests the neuronal excitatory network. Are we exciting the neurons when we expose them to citicoline or Cognizant as compared to placebo? And then the second part was to take those cells and run it through RNA sequencing and look at what mRNA is being expressed as compared to placebo. So before we get into it, though, I think it's always good to kind of share what are these cells I'm talking about. So I think most people understand what neurons are, but microglia cells are immune cells, and they're I basically call them the immune cells of the CNS. Astrocytes are supporting cells to the neuron. They provide structural support, metabolic support, and neuroprotective support. So it's good to know what they are, and they're representing what brain tissue would be.
And real quick, that Triculture type of study, that's like a commonly accepted study. That this isn't something you invented, is it?
Yes, no, this is kind of I wouldn't say it's standard practice, but I would say that's emerging science is using more commonly. This is what we were advised to do, and this is where kind of, because it's really cutting edge type of science, so we're learning as we go, but this is the probably best case scenario to represent brain tissue.
Cool.
And so we did, in this case with the Citicoline excited neuronal network test, we did 3 different arms. We did the saline, which is represented in gray, so that's our control, and then we did Citicoline, which is in dark green, but we also did choline and cytidine. And I think this is a really cool part that I think everybody should understand, that when you take Cognizant, you get Citicoline, and we know for a fact by pharmacokinetic data there is more citicoline in your brain when you take Citicoline, but you also get choline and cytidine. So it shouldn't be ignored what exactly those components or those ingredients are doing in your body as well, because just basically when you take Cognizant, you get all 3. So it's cool. In this research, we shared and looked at all 3 and what could be happening on the body. So what you're seeing now on the left, the spike rate, is basically like excitement of the neurons. How excited are they? How much compared to saline? And this is spike rates per second. So basically like neuron excitement per second. Burst rate is how many neurons in the neuronal network we are actually exciting. So it's like how many excitement we're having, and then how many cells are involved. So you can see here both Citicoline and choline definitely outperformed placebo for both sides. So pretty cool. This was done over a 24-hour period. Maybe next time we'll do it longer, but for now we started with that. So now going into the second part of this, which is the transcriptomics part, looking at that mRNA sequencing. So interestingly, this was a 4-arm study, of course, because it's in vitro, so it's a little easier to do more. So what we did was Citicoline, we did the choline plus cytidine, and then we did a positive control, which is donepezil or Aricept. That's a drug. It's sometimes used for various CNS disorders like Alzheimer's. And then we did saline solution, it's called PBS, but it's phosphate buffered saline, just so you know. So we ran all 3 of these, all the tricultures were exposed to each one of these. So it's like 4 arms. And so we extracted the mRNA. So you have the untreated versus treated, and the treated could be, donepezil or Citicoline or the combination of choline and cytidine. And you run it through mRNA sequencing, and ultimately you go one at a time and you look at what genes were expressed in the treated cells versus the untreated cells. And that gives you differentially expressed genes. And those differentially expressed genes, there's a huge library that exists that tells you kind of what molecular pathways those genes regulate or are involved with. And then to take it a step further, there's also disease transcriptomics library, so you can then map, okay, this gene and then what disease is it linked to or associated with. We're definitely not going after disease claims here, but it is a great way to kind of understand where the next research, potential could be with Cognizant. So that was why we did that as well.
Gotcha. Now, not to totally interrupt, but all these slides say Thorne Health Tech. Was there a collaboration with Thorne Vitamins here?
Yes, I'm sorry I didn't mention that earlier. Yes, Thorne, Health Tech. We were partnered in this study together, and we used their labs, and we also used some of their partners as well for the research. So this is unpublished data. hoping to publish like early part of next year. So stay tuned for that. But yeah, we did it in combo.
Okay, thanks.
Of course. So here is the analysis. It's a lot. These are what we call volcano plots. And just follow me here for 1 second. I'm going to focus mostly on the citicoline versus the saline. So what we see here is anything that's orange dots are genes that were expressed by citicoline and not by the placebo or the saline. But on the left here of the V is the downregulated genes. On the right is the upregulated genes. So anything orange was expressed by citicoline and not by saline. And these dots represent the differentially expressed genes, and it becomes what's called citicoline's gene signature. So these are all the genes that it activates that, it wouldn't be active in a brain without citicoline. And the difference is about 749 genes. So it's a lot. And just so you know, this is a sneak peek because we haven't even been able to analyze all of it because it's so intense and complicated. So molecular metabolomics is extremely extensive, and I can't even. So stay tuned. I can't even go into these other volcano plots today. I don't have all that data yet. We're still working on that data, and I especially am really excited to look at the difference between citicoline and choline versus cytidine. What they did say at the presentation at ASN, some of the experts that presented this did say that the genes that citicoline turned on versus choline and cytidine were more related to brain function and structure and support, because choline and cytidine are, throughout the body and have a lot of different roles. So that was interesting, but more to come there.
Right. So just because one thing activates more genes or whatever doesn't necessarily mean that doesn't have to be a good thing all the time.
Right.
We want it to be the right genes.
Exactly. And that's what you're looking at. So here is, and this gets complicated, but I'll just kind of walk you through it. So ultimately, we haven't done an analysis of every single gene. That's going to be Katie's job next quarter, and possibly a little bit. So we're going to look at this. We're going to look at this closer, but generally speaking, a summary, these are the most differentially expressed genes for Cognizin as compared to placebo. And these were the upregulated, the ones up here are the upregulated genes. And what we saw, I would say, like, this is over summarizing it, but a lot of the genes that were represented here had a role in structure and function of the CNS. So the neuronal plate and the neuronal crest, which is the structure of this part of the structure of the CNS. So interesting there. And it actually also has to do with development. So it makes you wonder, like, could this be like a good ingredient for brain health development as well? And then at the bottom here is this gene that's pretty well known in the literature called NCS1. It's a calcium sensor gene, and basically it's down regulated by citicoline and donepezil aricept. And what we know is that this gene is involved in nerve cell terminal growth, memory, learning, but on the flip, it's highly upregulated in disease states like schizophrenia and bipolar. We don't know exactly why, but we do know when they do autopsies of schizophrenic and bipolar, this is overexpressed. So of course, we don't want to underexpress it either, but it's good to know. And of course, we're mimicking Aricept, which is, I guess, the positive control in a way.
Yeah, it's always tough to just look at that and make a decision on anything. You don't know if your body is if it's upregulating something as a rescue mechanism, or if it's actually an effect of I got a disease and then this bad thing happened. It could be two completely different things where it's good and protective versus a result of something bad happening. So it's always good to know that stuff, given what I almost like to think, given how much other beneficial stuff we've seen from Cognizin, I defer to the more healthy side of things in this case, but it's worth monitoring.
One thing I didn't say is all this could not be clinically relevant. Right?
That's true.
We think it is. We believe it is because we have a lot of benefit. We see a lot of benefits, right, in the literature, and we know, but doesn't mean that this NCS1 is clinically relevant, right? It means that this is all just telling where we do need to do a little more digging in terms of, is it connected to some of the benefits we already see, which is, like I said, some of the next steps, and or is this a benefit that maybe we want to test to see if it has clinical relevance in the future? So that's kind of what, but yeah, I'm not convinced all of this has any clinical relevance.
Well,
yet.
I mean, that's so cool. It's kind of hitting me right now how far along this is. We're not used to this type of discussion. A lot of times people are trying to demonstrate that their ingredient may or may not work. You're so far deeper into we know it works. We're going backwards back into the cells where, like generally you get into human and everyone's like got a couple good human studies and they're done and they're happy. We're going backwards back into the cells to see why it works better, to see if there's more benefits that we could like pop out of other human studies. So it's like, that's why this is different. And I like it because you're not just stopping like most companies would. So thanks for doing that.
Yeah. And we will use this. And just so you know, like this data was so good that at ASN, which has thousands and thousands of abstracts, we got like floor time to show the science. So this is pretty high level.
Sounds like
and
a fun
I'm not
show.
even the expert here, just so you know. So stick with me. All right. So what there is other also like these molecular pathway libraries where you can plug in like, okay, this is the gene and this is the molecular pathway it's involved in. This is very overarching, kind of bigger picture. We are going to work on drilling down on it a little bit more, but these are upregulated molecular pathway enrichments for citicoline. And ultimately what we saw was enhancing cholesterol synthesis, enhancing synapses, so that's when the neurons speak to each other, and neuronal development, so nerve cell development. So the cholesterol thing is really interesting, I think, because just to give you a little background on what happens there, and we've seen this in different preclinical studies as well with the cholesterol, is when we get our neurons die, but when they die, it kind of creates debris in our brain, and our brain is somewhat good at clearing it, but it also, if the neurons are dying at a faster rate as we age, unfortunately, there can be a lot of debris. And the cholesterol can kind of build up and it can create more, it can actually become neurotoxic and also prevent good blood flow. So upregulating cholesterol, it's called dyshomeostasis, like it's kind of like I call it the vacuum cleaner for cholesterol, is really a positive thing when it comes to like the brain health. So there's a little bit more on that to come too.
Okay, good.
All right, and then this is the down-regulated molecular pathways. So one thing that's interesting, and we did also see this in other data where we believe we decreased alpha-beta secretion. And you might know amyloid plaques are like the plaques that are involved with Alzheimer's and stuff, and amyloid secretase is the enzyme to make that alpha beta secretion. So we are down regulating that. Alone alpha beta secretions are neurotoxic. So this is implicated potentially in, some helping in certain people that could be more prone to amyloid plaques, or in general, it's just neurotoxic as well. So interesting. We're going to dig in there a little bit more for sure too.
Yeah, that's been a hot topic of discussion with the amyloid beta plaques because there's been some, there's a little bit of fraudulent research way long ago, but it definitely does seem like it is there in the Alzheimer's story. It's just, it's not shown to be, we don't know if it's causal or what. Yeah.
Reason, right? It's not where we did think the plaques themselves were the reason. Mm-hmm. Now we know it's not necessarily, but they're definitely involved in the process, right? So, So, and then this is notch signaling, which is pretty well talked about in the literature. There's multiple different types of notch signaling. It has to do with brain tissue development. It also has been shown to be a targeted therapy for anti-tumor cells and things like that. So we're going to dig in a little bit on that and see where we go with that. And then this is, like I said, the next step is they took it into a disease network library. So taking those unique differentially expressed genes, putting it into a disease library. Again, this we have to take with a grain of salt. Okay, what do we see? And there's more to come. This is just a quick kind of highlight. But what was really interesting is we saw a lot of disease popping up that had related to mitochondrial dysfunction. Mm-hmm. Mm-hmm. So what we think is going on here from some of the science is we believe there's there's an upregulation of a bioenergetic shift in the neurons in how they're metabolizing ATP. And we have that data. I can't share it today, but we can show that. So we do know we're enhancing the mitochondria's ability to produce ATP, but we also know that just like we enhance the cell membrane, we also can enhance the structure and function of the mitochondria. So ironically, a lot of the pathways that were found actually were triggered by diseases related to mitochondrial dysfunction. So that for me is kind of the next step, right?
Right. I was going to say, so if I had like $1 billion, the next idea is you're thinking maybe run a 500 milligram Cognizant study on women with PCOS. Is that what we're trying to like possibly learn here eventually?
Well, I think mitochondrial health has been connected to so many dysfunction, especially in brain health. We know that's a huge area. Definitely women's health is a hot button right now. Like everybody wants to know if we have science around that. So I don't know if we'll go after PCS overall or not. I do think we're going to look into women's health for sure. But I do want to dig into the mitochondria health a little bit more and health span and see if, understand. There's some other ingredients on the market that, seem to impact mitochondria health. So definitely going to, take a look and see where can we go next with this?
Yeah, given how Cognizant feels, I wouldn't be surprised if there's mitochondrial benefits. Like
for sure. Yeah. And I think the science has already pointed to it in a lot of ways. So it's just a matter of putting together something robust enough in a healthy population. That's always a challenge when you do next steps for studies is like, is it going to be effective in a healthy population? So yeah, so this is really cool science. We saw that we can increase the Citicoline gene expression signature increased the cholesterol metabolism, helping clear out cholesterol from the brain, and it helped with synapses, neuronal activities. It downregulated amyloid and Notch signaling. We're looking into finding out more around its signature to reverse mitochondrial dysfunction, maybe even lipid dysregulation conditions, considering. So we're going to dig in a little more on the NCS1 gene to see, okay, we know that, we're not looking at bipolar or schizophrenia, but we want to look at the positive impact of downregulating that gene and what can that be in a healthy population or for health span. And this is a pretty busy diagram, but I think it kind of helps like predict the mechanisms a little bit. So neuronal death is kind of the ultimate problem that happens as we age. So preventing neuronal death is the key. So this cholesterol dyshomeostasis is how we kind of prevent neuronal death, getting rid of that cholesterol. Metabolic capacity, so that mitochondria function is important, and then that AB secretion. AB secretion is implicated in Alzheimer's, mild cognitive impairment, and so so these is kind of putting it all together in a way, but it's a pretty busy diagram. And that's it. So next step. So for us, we hopefully can come back and share with you more on what we tease out, because as you saw with the volcano plots, we also did, comparison of choline and cytidine, but we haven't even been able to look at the differential expression of those two. But I really can't wait to see that data because it's not a bad thing. It's not a good thing. We get it all when you take citicoline, but I'm curious to know what the differences are. And then, there is some existing animal data as well on this, and we want to see if there's any overlapping data there. So we're excited. And then we're going to do a better job really looking at, the expression of the genes and what specific genes do, and really even maybe having like a clear metabolic pathway for each gene that gets upregulated. But the ones that are more expressed upregulated. So just so you know, going back to the volcano plot, the ones that are higher on the plot are the ones that are more expressed. So those are the ones of more interest to us when Katie's going to go through some of these genes and look at like what they're actually doing. We're first going to look at these ones. These ones that are the real outliers, because those are the ones that are the most expressed. So that's next.
Super interested in the choline and cytidine content, because I think we all believe in Cognizant. I think we all like choline in general. I think what sets Cognizant apart from other choline donors is one of the biggest selling points of Cognizant itself. So that's going to be interesting.
Yeah, and I've even like through the years, because I've been working for Kyowa forever, some people will be like, well, why can't you just take choline and cytidine then? But this data is going to show us really that it is different, right? Because when you take the citicoline, you actually do get citicoline in the brain as well, and there is a difference in what it does in the body, and this is going to tell us what it is.
So there's always a lot of controversy around cholesterol.
I know.
Because we see the data, and like a lot of things, I think there's a kind of a J-shaped curve on things or whatever, but it could be protective. It's a lipoprotein that carries a lot of stuff. Sometimes that stuff could be good. Sometimes that stuff could be bad, So, but in terms of, I've never really seen much about cholesterol in terms of brain health and cognitive function. Like, do you know much about anything there, or is it more of a general like cell?
Well, I guess the question I have is, I think we know that it helps when it comes to disease, because there's a lot of science around brain health trauma and stroke. And what we get there is excessive cell death. And we know citicoline can come in and rescue and clear out a lot of that debris. And basically, when you look at some of that science, it's kind of explaining, they think citicoline is almost like an antioxidant and clearing a lot of the debris. So I think it's related to that, but I don't know if in a healthy brain, what that looks like. And I do think it does matter what gene we're talking about with the cholesterol too, because there's a lot, right? So I want to dig into exactly what we're looking at there and really understand it a little bit more. But this is like preliminary, like first sight data, and we're still working through the science right now.
Mm-hmm. So yeah, back to the PCOS thing or something. And we talked about how women's health is a hot thing. If we were formulating a women's health supplement, and we did include Cognizant, because like I said, it's experiential. It's going to feel good. Mm-hmm. There are benefits. I don't really have a question. I'm just thinking. I like to formulate on these podcasts too. Like, we like to do something like out of left field. If you put in Cognizant into a women's health product, and you have other ingredients that can make the health claims on women's health, we could provide Cognizant could provide the claims for like brain health, basically. And then maybe there might be like a side benefit here. It's kind of a hope and a prayer and everything. But like, that's kind of like what I'm thinking is a possibility for someone who's looking to get creative.
Of course, my marketing is going to love what I have to say now, and they're going to try to make claims on it and everything. But the reality is, as estrogen declines, there is an adjustment to your brain. Your brain starts to go into withdrawal, and there is this like kind of fog, and there is a reaction that the cells have as the estrogen goes down. Mm-hmm. And it definitely, I believe there is some bioenergetic shifts as well. So we're actually, another thing Katie's doing is looking into kind of, what are the shifts that happen when our hormones change, and could, those changes be helped with some of the mechanisms that we have with citicoline, right? So that's how we're going to take a look at it to potentially see how we can impact. But I believe we can have a benefit. In fact, we did a study in healthy middle-aged women between the age of 40 and 60 for focus and attention, and it came out positive. And at the end of the study, the women didn't want to give it back. They really liked it. And we were just told that FYI. So, and we do believe it could have some impact on health, but we're still asleep, and so we have to still look into that, because what people expect from sleep is like, oh, it's going to make me fall asleep. Well, it's not necessarily going to do that. We know it doesn't create drowsiness, but we think it could help with quality of sleep.
Gotcha. Awesome.
We actually kind of typically don't take Cognizant around sleep, because it's not a stimulant, but it kind of is stimulating. It kind of, you might think that it might keep you up, but that's interesting.
Yeah.
Well, Ben, you've never used so high dose choline is sometimes used by people who are going for lucid dreaming before bed. So I don't know. I have actually not done it with Cognizant. That might be interesting, but sometimes the brain gets a little bit wired up, and if you can fall asleep, that
was kind of my
dreams
point.
Does happen.
Like, when we talk about Cognizant, we kind of say it's kind of like the stimulating choline. Like, it does kind of wake you up. So I've seen like other choline donors pre-sleep, but never Cognizant. So maybe I'll be trying that tonight.
Yeah, we don't know for sure, right? We do think there could be an impact, and again, I'm cautiously entering that, because I'm like, well, it's not going to be like a melatonin. Let's not pretend like we're going to have a melatonin impact, because we're not. But I think that people might wake up feeling more rested, because they got better quality of sleep potentially. But we got to do that, right?
Now, this type of study, another ingredient of yours that I'm thinking might make a lot of sense is Cetria. Well, like, did you get enough out of this study to want to do this type of study on other ingredients?
Absolutely.
Okay, because I'm saying Cetria is L-glutathione.
Yeah.
It's not just L-glutathione. It's you could probably explain it better than me, but like there's so many things that it might upregulate genetically.
Yeah, I mean, I followed the research on glutathione because we have an ingredient, and I can't even keep up
Right.
with the literature that's getting published on the molecular pathways that glutathione impacts. So it's a lot to follow.
Do you get like PubMed alerts on a lot of
Yeah.
things? Oh, okay. Yeah.
It's like it just doesn't stop. And you read the titles and you're like, what?
Right.
So there's a lot of science we already know, and I think a lot of people are already doing a lot of the work there. But definitely, this is a different spin on it too.
Awesome. Okay. Well, we kind of have everything here. I'm excited. Are you going to be at Supply Side West?
I'm not, but Katie will be.
Oh, all right. Well,
I had a conflict this year, but I'll be there in future years.
Oh, you didn't want to leave for Halloween, did you? I'm leaving early that Thursday morning, so there's only so many 7-year-old Halloweens you have in your
That's
in your life.
exactly my point. I have a 7-year-old too.
Oh, there we go. Me too. Yeah.
7 and a 9-year-old.
Yep. So yeah, leave it early, but either way, yeah, we'll be excited we're gonna have a lot going on with you and everything. So this will publish before then. So following you, do you do anything on social media? Are you active on LinkedIn or anything like that? Do you want us to post your profile, or is it mostly like Kyowa Hakko USA stuff?
Yeah, like my social media is personal at this point. I'm not. That's why we hired Katie. Like, I'm not interested. I'm like old, I guess. Like just not interested.
I think it's a totally reasonable thought process.
Yeah.
But I mean, people don't have to follow you then, but this to me seems like a fun jumping off point for where things can go from here. I mean, I think we all kind of accept Cognizant as kind of one of pretty much our favorite choline donor right now, but there's a lot of forward direction to go from here.
Yeah.
So it's exciting.
Well, just so you guys know, and because, we're partnered with you, so we are actually in the works with one study for reaction time. So we have a patent on reaction time, but it's all preclinical data. So now we're doing a human study to see if we can get that reaction time, and then we have patented benefit. And you guys know there's another patent coming. It's not ready yet, but we can share that. Maybe Katie could share that patent coming out pretty soon.
Are we okay to publish what you just said? We're still recording. I mean, we could edit it out, but...
Yeah. So we are working on, we have a patent, but we're working on human science to hopefully, really have that patent. So the future is, because we are a brand and we're investing in this research, is to really patent the benefits that we're investing in the research on, right? So we can differentiate ourselves in the marketplace. We don't want people, piggybacking on our science.
Well, I think for now, I think you guys are doing pretty good at that. I think you've both done a great job of producing data, but also communicating what makes Cognizant different. So
good, I hope so. So I didn't even finish. So we're doing a motor speed study that's already almost started, and then we are doing, and this is between us. So now Katie's planning a mood study right now. So we're looking at POMS, and then we're going to look at an acute study.
Cool.
So we're going to look at really acute data, but that's a pilot, and then we'll go further to, if the acute pilot comes out, we'll go on to do more acute.
Did you say you're looking at POMS?
POMS is like a
POMS.
Yeah,
right?
it's a standardized test that measures mood. It's a better test for healthy subjects to measure mood.
Okay, I'll know it better when I do the video for that.
Yeah,
I thought you were making a joke about reading POMS.
No, profile of mood states.
Yeah.
Okay, got it.
Exactly.
I promise I'll know the test more when I talk about it professionally.
Sorry about that. I just say these things and I forget that it's not.
I should know about it, so it's all good.
Yeah.
Awesome. Well, Danielle, thank you for joining us and keeping us up to date on everything you're doing. I love that you're really drilling down this deep. So we do appreciate it.
Absolutely. Thanks for being a good partner with us too, and have fun at Supply Side.
Thank you.
Thanks.
All right.